All right, welcome back to the deep dive. Today we're diving into the world of, well, how we even know what's making us sick in the first place. Diagnosing infectious diseases.
Yeah, it's a whole detective story happening at the microscopic level, isn't it?
It really is. And it all starts in the lab. We've got a whole chapter here packed with info on how those clinical microbiology labs identify the culprits and figure out how to fight back. So consider this our deep dive into that knowledge.
Think of it as a guided tour through the essential techniques and the whys behind them, really getting into the nitty-gritty of how we pinpoint those tiny invaders causing all the trouble.
Exactly. Our source material lays it all out in three main sections. First up, we're going behind the scenes in the microbiology lab itself.
Safety first?
Absolutely.
Huh.
Then we'll explore the classic techniques, the tried and true methods of isolating and characterizing those microscopic bad guys.
The foundation of it all.
For sure. And finally, we'll get into the really cool stuff, the cutting-edge immunological and molecular tools that are changing the game of disease diagnosis.
It's moving so fast.
It really is. Ready to jump in?
Let's do it.
Okay, part one, the microbiology lab and the healthcare environment. Safety protocols front and center, right?
Absolutely. You're dealing with a whole range of microorganisms, some pretty nasty and resistant to multiple drugs.
Multi-drug resistant pathogens, we've all heard about those.
Exactly. So safety in these labs is paramount. The chapter really drives this home. Strict adherence to protocols isn't just a good idea, it's essential for protecting lab personnel.
And preventing the spread of infection.
Of course, it's about safeguarding the whole diagnostic process.
So what are we talking about specifically? PPE, I imagine. That's the first line of defense.
Right, your standard lab coats, gloves, eye protection, face masks, all playing a role.
Each piece has its own job to do in minimizing exposure.
Exactly. And the chapter emphasizes that just wearing them isn't enough. You've got to use them correctly, consistently.
It's all about proper training and habits.
Yeah, and there are other key safety rules. Limiting access to the lab, strict hygiene, think hand washing, lots of hand washing.
Can't emphasize that enough.
Making sure lab workers are vaccinated against relevant pathogens, handling patient samples carefully, even something as simple as laundering lab clothes regularly, it all contributes.
Makes sense. It's a whole culture of safety. You know what really struck me, though? The chapter points out that most lab infections, accidental ones, don't happen from big dramatic spills or anything.
Oh, really?
Yeah, most of the time it's routine handling of specimens or, get this, infectious aerosols.
Those tiny particles in the air.
Yep, we don't even see them, but they're a real risk factor.
It really highlights the constant vigilance needed, doesn't it?
It does. And speaking of risks, that brings us to HAIs, healthcare-associated infections, sometimes called nosocomial infections, infections you pick up in a healthcare facility.
A big concern. The chapter says about 10% of hospitalized patients in the U.S. get an HAI. That's huge.
10%. So what makes someone more susceptible to getting an infection while they're already in the hospital?
Well, if you're already sick or have a weakened immune system, you're naturally more vulnerable. Very young patients, elderly patients too, their immune systems aren't as strong.
Makes sense.
Then you have proximity to others who are infectious, of course. And sometimes healthcare workers themselves can be carriers even without symptoms.
That's a bit unsettling, isn't it?
It is, but it's a reality. And then any medical procedure that breaks the skin barrier, surgery, catheters, even just an IV line, those can be entry points for microbes.
It's all about minimizing those risks.
Exactly. And some treatments, like anti-inflammatories or antibiotics, they can actually disrupt the body's natural microbial balance, potentially making a patient more susceptible.
It's a delicate balance for sure. So any examples of the usual suspects behind HAIs?
Oh yeah, we've got the usual suspects, Staphylococcus aureus, including the antibiotic-resistant MRSA, Enterococcus, Escherichia coli.
We hear about those all the time.
Right. And Klebsiella pneumonia, Acinetobacter, and the particularly tricky one, Clostridioides difficile. They've often developed resistance to multiple antibiotics, making treatment tough.
So how do we prevent these HAIs? It seems like a team effort is needed.
It definitely is. The chapter stresses that everyone in the facility, from the infection control team to every single staff member, plays a role.
Everyone's responsible for infection control.
Right. You need to assess patients right away to see if they need isolation, screen healthcare personnel for any asymptomatic carriers, and of course adhere to standard infection control procedures all the time with every patient.
It's about creating a culture of prevention, right?
Absolutely.
Healthcare facilities, by their nature, they bring together a lot of sick people or people with weaker immune systems. It's a high-risk environment by default.
It is. And to manage those risks, especially in the lab where they're working with these pathogens directly, there's a system of biological containment.
Low safety levels, right?
BSLs. Exactly. It's a hierarchy based on how dangerous the pathogens are and how much containment they need.
So from least to most stringent.
From BSL-1, the lowest risk, up to BSL-4, the most dangerous. But no matter the level, everyone has to follow standard good laboratory practices. That's the baseline.
The foundation for everything else. So where would you typically find these different levels?
Well, BSL-1 and BSL-2 are pretty common. BSL-1 labs you'll find in colleges, universities, working with organisms that are generally not a big threat to healthy adults. Standard clinical labs where most diagnostic work happens, those are usually BSL-2.
And BSL-3.
BSL-3 is more specialized. You'll find those in major clinical centers, research institutions. They're dealing with agents that could cause serious or fatal disease through inhalation.
It's serious.
It is. Then you have BSL-4, the highest level of containment. These facilities are for the really dangerous stuff, pathogens that are highly likely to cause life-threatening disease, and there might not be any vaccines or treatments available.
That sounds like something out of a movie.
It kind of is. The CDC, the U.S. Army Medical Research Institute of Infectious Diseases, USMRID, those are the kind of places with BSL-4 labs. They need total isolation to prevent any release of those pathogens.
It's really fascinating, the level of precaution needed.
Okay.
So that's the lab environment. Let's move on to part 2. Isolating and characterizing those infectious microorganisms. This is where the detective work begins.
Right. Someone comes in with symptoms, and the question is, what exactly is causing it? You've got to pinpoint the specific microorganism.
Like finding a needle in a haystack sometimes, I imagine.
It can be. The chapter explains that it all starts with getting the right sample, tissue, or fluid from the infected area.
And keeping it clean, right? No contamination.
Absolutely. That's crucial. Proper specimen collection is key to accurate diagnosis.
So what are the big things to consider when collecting a sample?
Well, it has to be aseptic, no contaminating microbes from the environment getting in.
Sterile technique.
Exactly. And the sample has to be big enough to run all the tests.
Not too small.
And you've got to keep the suspected organism alive. Some need oxygen, some don't.
So many factors to think about.
Right, and then get that sample to the lab and process quickly so nothing degrades or grows too much where it shouldn't.
So time is of the essence.
It really is. The chapter mentions sterile swabs. That's a common way to get samples from wounds, the nose, the throat.
Makes sense, those are pretty accessible.
But for deeper infections, you might need blood draws, a lumbar puncture for cerebrospinal fluid, even biopsies during surgery.
So it depends on the location of the suspected infection.
Right. And once you have the sample, there are direct detection methods to get a first look.
I remember learning about gram stains. Is that one of the first things done?
It is. Looking under a microscope, especially with a gram stain, can give a quick preliminary diagnosis.
A quick way to classify.
Yeah, it differentiates bacteria based on their cell wall structure. Gram-positive bacteria, they hold on to that crystal violet dye. They look purple. Gram-negative, they lose that first stain, but take up a counter stain. They'll look pink.
So a quick visual clue.
Exactly. It can really guide early treatment decisions while you're waiting for the more definitive culture results.
Now, the chapter talks about specificity and sensitivity when it comes to diagnostic tests. What exactly do those terms mean?
So specificity is all about a test's ability to identify a specific pathogen, and only that pathogen. You don't want it giving a positive when it's not really there. Like a lock and key, only the right key opens the lock. That means fewer false positives.
Okay, I get that. So no false alarms.
And sensitivity, that's about the test's ability to find even tiny amounts of a pathogen. Like a super sensitive smoke detector, it'll pick up even the faintest trace of smoke.
Yeah.
Fewer false negatives, so you're not missing an infection that's actually there.
So you catch it early. Both are important. But I guess depending on the situation, one might be more critical than the other.
Exactly.
So after the microscope work, what's usually next in pinning down the culprit?
Often it's growing the pathogen in the lab. We call that culturing, and that's where different types of growth media come in.
I remember those Petri dishes from science class.
Yeah, those. So you have selective media. It's designed with substances that stop some microbes from growing, but let others thrive.
Like a filter, only letting certain things through.
Right. It helps isolate the organism you're looking for from a mixed crowd.
And then there's differential media. It doesn't stop growth, but it helps you tell different bacteria apart.
Exactly. It uses specific ingredients, so different bacteria show up differently, maybe different colors or changes in the surrounding media.
Okay, some examples.
Sure. Blood agar, that can show if bacteria can break down red blood cells. Chocolate agar, that's for those picky organisms that need extra nutrients to grow. Eosin methylene blue agar, or EMB, that's good for differentiating gram-negative enterics. And E. coli, it actually gets this metallic green sheen on EMB. Pretty cool to see. Then there's McConkey agar. That one's both selective and differential. It favors gram-negatives and also shows whether they ferment lactose. So you see pink colonies for fermenters and colorless for non-fermenters.
So much information from just a color change.
Exactly. And then you have enrichment cultures. Those are for when you think a particular pathogen might be there, but maybe in very low numbers.
So they're hard to find among all the others.
Right. You use special media and conditions just to give that target pathogen a boost. Let it grow enough so you can actually detect it.
Smart. What about for liquid samples like blood or cerebrospinal fluid? The chapter mentions automated culture systems. How do those work?
Those are great for constantly monitoring liquid samples for any signs of growth. They have sensors that pick up on changes as microbes multiply.
Changes in what?
Well, it could be the turbidity, how cloudy the sample gets, or maybe the microbes release gases or produce fluorescent compounds as they're metabolizing stuff.
It's all about detecting those subtle signs of life.
Right, and it's much faster than traditional methods.
Makes sense. So let's talk about specific specimen types. Blood and CSF. We mentioned those automated systems, incubating them with and without oxygen. And the chapter emphasizes that healthy cerebrospinal fluid should be sterile, no microbes at all. UTIs, urinary tract infections, those are super common, right?
Very common. First, they check the urine under a microscope, looking for white blood cells, bacteria. Then they culture it, usually using selective and differential media for those enteric bacteria that often cause UTIs.
And what about fecal cultures? That seems like a whole other challenge. There are so many microbes in the gut normally.
It's a whole ecosystem in there, so handling those samples is extra careful. You need a variety of media, some selective, some differential, to isolate the pathogens you're looking for.
So you can pick out the bad guys from the crowd.
Exactly. You might be looking for nasty strains of E. coli, like O157.H7 or Campylobacter. They can cause some pretty serious intestinal issues.
And wounds and abscesses, it sounds like there could be a real mix of bacteria in those.
You've got your aerobes, like Staphylococcus aureus, maybe Pseudomonas aeruginosa. Then you might have facultative anaerobes, those enteric bacteria that can live with or without oxygen. And then true anaerobes, they only grow without oxygen.
So you need different conditions to grow all of them.
Right, you culture them with and without oxygen to cover all the bases. The chapter also talks about chromogenic agar. That's a special media that makes MRSA colonies turn a specific color.
So you can spot them right away.
Exactly. Makes identification much easier.
And then for genital specimens, those are important for diagnosing STIs, sexually transmitted infections. I know for gonorrhea, they use a gram stain and a special media called modified Fayer-Martin agar, or MTM.
Right. Neisseria gonorrhea, the bacterium that causes gonorrhea, it's a bit finicky. It needs specific nutrients and conditions to grow.
The diva, basically.
You could say that. MTM agar is formulated to give it what it wants while also inhibiting other microbes that might be in the sample.
So it's a pampered environment for the gonorrhea bacteria.
Pretty much.
We talked about anaerobic pathogens earlier, the ones that can't tolerate oxygen. How do you culture those? Sounds tricky.
It is. You need special media, often with extra organic stuff, and reducing agents to keep the oxygen levels down. And the technique has to be really careful, minimize any oxygen exposure. They often use anaerobic chambers or containers for that.
Sounds complicated.
It can be.
So once you've identified the pathogen, you need to know what drugs will kill it, right?
Yeah.
That's where antimicrobial susceptibility testing comes in.
Absolutely. You need to know what weapons will work against this particular enemy.
So the doctors can prescribe the right treatment.
Exactly. And there are several key methods for that.
Starting with the minimum inhibitory concentration, the MIC. What does that tell us?
The MIC is the lowest concentration of an antibiotic that's needed to stop that specific microbe from growing in the lab.
So how much drug do you need to stop it in its tracks?
Exactly. A lower MIC means the bacteria is more susceptible to that drug.
It's more vulnerable.
Right.
Yeah.
You can figure out the MIC with broth dilution, basically testing different concentrations of the antibiotic in liquid media.
Trial and error, in a way.
Sort of. And there are automated systems that can do this much faster.
Technology's speeding things up.
Definitely.
Yay.
Then there's the disc diffusion test. That's a classic. You've probably seen pictures of those Petri dishes with the antibiotic discs and the clear zones around them.
I think I have, yeah.
So you put these little paper discs soaked in different antibiotics on a plate covered with the bacteria. The antibiotic spreads out from the disc creating a gradient
Gradient of what?
of the antibiotic. So you have high concentration near the disc, and it gets lower further away. If the bacteria is susceptible to the antibiotic, you'll see a clear zone around the disc where the bacteria couldn't grow.
It gets killed off by the drug.
Exactly. The size of that zone tells you how susceptible the bacteria is to that particular antibiotic.
So bigger zone means more susceptible.
Usually, yes. There are standard charts that help interpret those results.
The chapter also mentions the epsilomer test, the E-test.
Yeah, that one uses a plastic strip with a gradient of the antibiotic on it.
So it's like that disc diffusion, but more precise.
Exactly. You put the strip on the plate with the bacteria, and after it grows, you get this elliptical zone of inhibition. Where the bacteria stops growing tells you the MIC, the minimum inhibitory concentration.
So a direct reading.
Right. And then finally, we have antibiograms.
Those are like summaries of antibiotic resistance, right?
Exactly. They compile susceptibility data for all the important bacterial isolates in a hospital or region.
So you know what's resistant to what in that particular area.
Right. It helps doctors make better decisions about initial treatment, even before they know the exact susceptibility of the bacteria causing the infection in a specific patient.
So it's about making informed guesses based on the local trends.
You could say that. And it's really important for tracking resistance patterns over time so hospitals can adjust their treatment guidelines.
So it's a dynamic tool, constantly evolving.
It has to be.
Okay, so we've covered how to find the microbes and figure out what drugs will kill them. Now, part 3, immunological and molecular tools for diagnosis.
Now we're getting into the really cool stuff.
I know, this is where it gets really cutting edge.
This section is all about going beyond those traditional culture methods, looking at faster, more sensitive, and more specific ways to diagnose.
And it all starts with immunoassays.
Right. Immunoassays use the very specific binding between antibodies and antigens. You can design them to find the pathogen itself or stuff it produces.
So you're looking for the pathogen's fingerprints, basically.
That's a good way to put it. And you can also use them to track the patient's immune response, see what antibodies their body has made.
So you see how your body is fighting back.
Exactly. Serology is a big part of this, looking at antigen-antibody reactions in the lab.
Okay, so serology involves analyzing blood samples, right?
Right. You're looking for those antibodies the body's made against the infection. The chapter mentions antibody titer. That's the highest dilution of your serum that still reacts with a specific antigen. A high titer could mean a past infection or exposure.
So your body has seen this before.
And if the titer goes up between two samples taken at different times, that's a strong sign of an active infection.
So your body's currently fighting it.
Exactly. The chapter also mentions skin tests, like the tuberculin skin test for TB.
Right, those are different from blood tests. They're done right on the skin, aren't they?
They are. They look for what we call delayed type hypersensitivity. So in the tuberculin test, you inject a bit of the TB antigen under the skin. If you've been exposed to Mycobacterium tuberculosis before, you'll get a reaction, some swelling and redness.
So it means your immune system recognizes the TB bacteria.
Exactly. It's already primed to fight it.
Okay, let's talk about monoclonal antibodies, MABs. We hear about those a lot for treatment, but they're also important in diagnostics, right?
Absolutely. The cool thing about monoclonal antibodies is that they come from a single clone of antibody-producing cells.
Because they're all identical.
Exactly, which means they're super specific. They'll only bind to one very precise target, and that makes them super valuable for both diagnosis and treatment.
So they're like guided missiles targeting only the bad guys.
It's a good analogy. The chapter explains how they're made. It involves fusing a B cell, the kind that makes antibodies, with a myeloma cell.
What's a myeloma cell?
It's basically a cancerous plasma cell.
So it replicates uncontrollably.
Exactly. That fusion creates what we call a hybridoma, which is a cell that can continuously produce large quantities of that specific antibody.
So you've got this factory churning out these highly specific antibodies.
That's a good way to put it.
The chapter then gets into different types of visible antibody reactions used in diagnostics. Starting with precipitation, what is that all about?
Precipitation happens when you have soluble antibodies in a sample, and they bind to soluble antigens. If the amounts are right, they'll clump together and form this insoluble complex that you can actually see.
So it falls out of solution.
Exactly, a precipitation.
It's like when you mix two liquids and you get a solid forming.
You got it. Immunodiffusion tests, often done in agarose gels, use this reaction to detect specific antibodies, for example, in diagnosing some fungal infections.
So you can literally see the reaction happening.
You can. Then there's agglutination, which is basically clumping.
Ah, like that rapid strep test they do in the doctor's office?
Exactly. That's a perfect example. So in agglutination, the antibodies bind to antigens that are attached to something, like cells or little latex beads, and that causes those particles to clump together, which you can see.
It's like they're all holding hands.
In a way, yeah. So in that strep test, the latex beads are coated with antibodies that specifically bind to the strep bacteria. If it's in your sample, boom, you get clumping.
Easy to see. And I remember another example, hemagglutination. That's when red blood cells clump together.
Right, that's how they do blood typing.
Okay, then there's immunofluorescence. That one sounds really cool, making the pathogens glow under a microscope.
It's pretty neat. You take antibodies and attach fluorescent dyes to them.
So you can see them light up.
Exactly. You use those labeled antibodies to probe a patient sample. If the target antigen is there, say, on a bacteria, the antibody will stick to it.
And under a special microscope.
It glows. The dye emits light at a specific wavelength, so you see these bright spots against a dark background.
Awesome.
There are two main ways to do it, direct and indirect. In direct immunofluorescence, the primary antibody, the one that binds the target, is labeled with the dye.
Straightforward.
Right. In indirect, the primary antibody isn't labeled, but then you use a secondary antibody that binds to the primary antibody, and that one has the dye.
So it's like a chain reaction.
Exactly. It amplifies the signal. This technique is used for a whole range of microbes, bacteria, viruses, fungi, parasites.
Really versatile.
It is.
Okay, moving on to enzyme immunoassays, or EIAs. That includes the ELISA tests, which are pretty common in labs.
They are. They're highly sensitive and specific. The key is that they use enzymes linked to either the antigen or the antibody.
Okay, enzymes are those proteins that speed up chemical reactions, right?
Exactly. So when you add a specific substrate, the enzyme will make a reaction happen that creates a detectable signal, usually a color change. And how intense that color is tells you how much of the target antigen or antibody was in the sample.
So you get a visual readout.
You got it. There are different types of EIAs, direct, indirect, sandwich, and combination, each with its own purpose.
And then there are those rapid tests, the ones you can do right in the doctor's office.
Right. Those are like simplified EIAs. They have all the reagents on a strip or a membrane, so they're easy to use and give quick results.
Great for when you need an answer fast.
Exactly.
The chapter also talks about immunoblots, sometimes called Western blots. I think those are used to confirm results from EIAs, especially for HIV.
You're right. They're more complex, but also more specific than many EIAs. You start by separating the proteins in a sample by size using something called gel electrophoresis. Then those proteins are transferred to a membrane.
So you're basically sorting the proteins out.
Right. And then you use specific antibodies that bind to the proteins you're looking for. If those proteins are there, the antibodies will stick.
So again, it's about that specific binding.
Exactly. Then you use another antibody that binds to the first antibody, and that one has a label so you can visualize it.
So the two-step process.
Right. You end up with these bands on the membrane that correspond to the proteins from the pathogen, like a fingerprint.
A unique pattern.
Exactly. Very specific.
Okay, final stretch now. Nucleic acid-based clinical assays.
This is where things get really exciting. PCR is the big star here.
PCR, polymerase chain reaction.
Yeah, it's revolutionized how we identify pathogens.
Now, how does it work?
It amplifies specific DNA or RNA sequences that are unique to a particular microorganism.
So it's like making millions of copies of a tiny piece of the pathogen's genetic code.
That's a good way to think about it. It's so sensitive you can find even the tiniest amount of a pathogen's DNA or RNA in a sample.
Amazing. And the chapter also mentions nucleic acid hybridization.
Right, that uses labeled probes that have a sequence that matches the pathogen's DNA or RNA.
So it's like finding a specific word in a huge book.
Exactly. And if that sequence is there, the probe will bind to it, and you can detect it.
And there are different types of PCR, right? Like qPCR and RT-PCR.
Yep, qPCR, or quantitative real-time PCR, it uses fluorescent probes to track the DNA as it's being amplified.
So you see it happening in real time.
Exactly. And not only can you detect the pathogen, but you can also measure how much of it is there.
So you know the severity of the infection.
Exactly. RT-PCR, reverse transcription PCR, that one's specifically for RNA viruses.
Because regular PCR works on DNA, right?
Right. So for RNA viruses, you first use reverse transcriptase to turn that RNA into DNA, and then you can do regular PCR.
So it's a two-step process.
Yep. And then there's qualitative PCR, which uses labeled primers within the amplified DNA, sometimes with melting curve analysis.
Melting curve analysis, what's that?
Well, DNA strands separate at a specific temperature, and that temperature depends on the DNA sequence. So by carefully measuring the temperature at which the DNA melts, you can tell even very similar pathogens apart, like herpes, simplex virus, type 1 and type 2.
Wow, that's impressive.
It is.
The chapter points out that these nucleic acid tests have some big advantages over traditional cultures. They're much faster, and they can detect organisms that are really hard to grow in the lab.
Absolutely. PCR and other amplification methods can give results in hours, sometimes even minutes, compared to days or weeks for cultures. And they can find those stealthy microbes that don't like to grow in our artificial lab conditions.
So it's revolutionized diagnostics. But there are limitations, right?
Of course. Finding DNA or RNA doesn't always mean the organism is alive and causing trouble.
It could be dead, but its genetic material is still there.
Exactly. And these tests don't usually tell you about antibiotic susceptibility.
So you still need those other methods for that.
You do.
Well, this has been an incredible deep dive into the world of diagnosing infectious diseases. We've gone from the basics of lab safety to the complexities of microbial cultures to the mind-blowing world of molecular diagnostics.
It's a fascinating field.
It really is. It's amazing how much goes into figuring out what's making us sick, all that work going on behind the scenes.
It's a testament to how much we've learned about the microbial world and how it impacts our health. And the tools and techniques are constantly evolving, which is exciting.
Absolutely. The shift towards these rapid and precise molecular methods, it makes you think how much faster and more accurately we can diagnose and treat infections now compared to even just a few years ago. And that has a huge impact on individual lives and public health as a whole.
It really does. It's a field that never stands still, always pushing the boundaries of what's possible.
Well said. We've covered all the key points from this chapter, from ensuring a safe lab environment to using the latest molecular tools to track down those microscopic culprits.
We've done a thorough job.
We have. Thanks for joining us on this deep dive into the fascinating world of infectious disease diagnostics. Until next time.